Hello, everyone, and welcome. My name is Rula Deeb. I'm a senior principal at Geosyntac Consultants in Oakland, California, and the coordinator of the webinar series on behalf of SERDP and ESTCP. Today's webinar will consist of a brief overview of both programs, followed by technical presentations featuring DoD-funded research efforts on improving the understanding of Pfas ecological risks. First, Dr. Mary Kurtz from the Academy of Natural Sciences of Drexel University will discuss the pathways and rates of Pfas uptake, bioaccumulation, and biomagnification within freshwater food webs. Marie's presentation will be followed by a brief Q&A session. Then we'll move on to Dr. Jamie Suskey from EA Engineering, who will talk about a tiered approach for assessing Pfas risk to threatened and endangered species. Jamie's presentation will be also followed by a Q&A session, and we will conclude the webinar with a longer Q&A session featuring both of today's speakers. The next several slides provide instructions on optimizing your webinar experience. If you have not done so already, please download Zoom at the link shown here and also provided to you in your webinar registration confirmation email. If you're unable to download Zoom, you can view the slides using a compatible internet browser such as Firefox, IE, or Edge, or by creating a free Zoom account. If you continue having difficulties with the slides, or if your screen freezes, try keying in Control and F5 to perform a hard refresh. If you are accessing the audio through your computer, click the arrow next to the Join Audio button, select Test Speaker and Microphone, and follow the prompts as they appear on your screen. If you continue to have difficulties, call into the conference line shown here. You can also submit a comment using the chat box. Please use the chat box only for comments related to technical difficulties. The Q&A option should be reserved for questions for today's speakers. And if you continue to have technical difficulties, just download a PDF of the slides from the webinar webpage and call into the conference line provided to you in your webinar registration confirmation email. Note that we will also be live streaming the webinar on the SERDP and ESTCP YouTube channel at the link shown here. So that's another option for viewing the webinar live. Today's broadcast will be listen only. You may submit questions by using the Q&A box on your screen. You do not need to wait until the Q&A sessions to submit your questions. We do encourage you to submit them well in advance of the sessions, and when you do submit them, please make sure to add your organization name at the end of your question so that we can identify you during the Q&A sessions. With that, I would like to introduce Dr. Andrea Leeson, who is the Deputy Director of SERDP and ESTCP, as well as the Program Manager for the Environmental Restoration Program area. Dr. Leeson has been with SERDP and ESTCP since 2001, and before that, she was a research leader at the Battelle Memorial Institute, where she conducted scientific research on the in situ bioremediation and the design and implementation of innovative biological, chemical, and physical treatment technologies for site remediation and industrial wastewater. Andrea received her doctoral degree in environmental engineering from the Johns Hopkins University. Andrea, please proceed.
Great, thank you, Rula, and thank you to everyone who joined us today for this webinar. Before we get into our lead presentations for the day, I just want to give a little bit of an overview about who we are and how this webinar series is fitting into the SERDP and ESTCP programs. So we are two companion programs. We are within the Department of Defense, and we're the environmental technology programs for the Department of Defense. And really, just briefly, SERDP is our science and technology program. So this is really traditional R&D. It is a partnership not only with the DoD, but also with the Department of Energy and the Environmental Protection Agency. And we've been doing this for quite a while now. ESTCP, the Environmental Security Technology Certification Program, is the companion program, and under this one we called our DEMVAL, or the Demonstration and Validation Program. And we take technologies or knowledge that's developed under SERDP, and once it's ready, we transition to the field to do small-scale studies to demonstrate and validate those technologies. It doesn't have to come from SERDP, but that is frequently a route that technologies go through, that they start under SERDP and develop into ESTCP. And very important to us under ESTCP is the technology transfer, that the knowledge we've learned in these pilot scale demonstrations can be transitioned to end users to better help with environmental management. So we have two main drivers for the sort of work that we fund. The first one is the sustainment of our testing and training ranges, as well as facilities and operations. And this is just a very high-level overview of the sorts of things that covers. And you can see it's very broad, from maritime sustainability, noise issues, forward operating bases. We deal with UXO and munitions constituents, as well as changing climate. So that covers a huge amount of issues for the DoD. We also are looking at reducing current and future liability. Now that can take the form of traditional pollution prevention, where we're looking to eliminate hazardous materials or hazardous processes, DoD installations. And then as well, we're looking at dealing with contamination from past practices. That can be impacted groundwater, soil, sediments. Our UXO liability falls under here. And then we're always looking for emerging chemicals of concern as well, so that we try to stay ahead of what we, what we know. And what really falls into this area significantly is our work on PFAS. And I put this up here not to go through it in any detail at all, but really to highlight this link at the bottom. And this link at the bottom, I will put the link in the chat room after I get done speaking. This is an interactive map of PFAS work, and each of these boxes show the title of a group of projects that have been funded. And we started in 2011, going out now till 2022 in terms of what we've solicited. You'll click on a box, it'll take you to another page that will show you all the projects that were funded under that solicitation, and every project there is linked to their project webpage, which will give you a little bit of an overview. So the presentations we are going to hear today fall under our PFAS effort, and I just wanted to give you a little bit of an overview of everything in terms of where you can get more information on these projects. As I said earlier, the technology transfer is extremely important to us. This takes the form of videos, traditional guidance and manuals. We do a lot of in-person training, and then we also do these webinars, which are a substantial part of our technology transfer effort. If you go to our website, you can see the list of all the different webinars that we have given, and this is just a list of some of the upcoming webinars that are available. And if you go to our website here, you can register for upcoming webinars, but also everything we've done in the past is archived, so you can go back and listen to the ones we have coming up. So at this point, Rula, I will turn it back to you to introduce our speaker.
Thank you so much, Andrea. It is my absolute pleasure to introduce our first speaker, Dr. Marie Kurtz, who is a senior scientist at the Academy of Natural Sciences of Drexel University in Pennsylvania. Her research addresses the transport and transformation of chemical constituents in freshwater systems, with a focus on biological processes and human activities that these dynamics reflect. In addition to research, a large component of her work involves synthesizing her own and other research findings into diagnostic tools and guidance to support the effective management and restoration of stream water quality and ecosystem integrity. Marie earned her bachelor's degree in geology from the College of William and Mary in Virginia, and her doctoral degree in geology from the University of Florida. Marie, please get us started with today's first technical presentation.
Great, thank you, Rula, and thank you, all of you who are listening in today. So for my presentation, I'll be starting with an overview of what we know about PFAS dynamics in freshwater systems, leading into some of the critical unknowns that still remain, of which there are many. From there, I'll introduce the objectives and approach of the current SERDP-funded study by myself and colleagues focusing on freshwater food webs, and end with how our results will be relevant to the DoD. Just a little spoiler at the very beginning, I'll admit that our major results have been significantly delayed due to COVID, so this will be more of an introduction than a thorough review of results today. Hopefully you'll tune back later to hear more how the study works out. So we know that there are, excuse me, let me start over. Per and polyfluoroalkyl substances, or PFAS, have been ranked amongst the highest priority contaminants of emerging concern due to their persistence, their bioaccumulative potential, their human and ecosystem toxicity, and their global distribution. There are many pathways of PFAS to the environment coming from sources including manufacturing sites, the disposal of consumer products, and firefighting foams used at airports and military sites. For today's talk, I'll be focusing on PFAS contamination in aquatic environments, in particular that associated with the historic use of aqueous film-forming foams, or AFFF. PFAS originating from the application of AFFF in terrestrial environments enters surface water streams through two major pathways. The first via stormwater runoff of contaminated soils, and secondly, from leaching from soil to groundwater, which then contributes to stream flow. Once in the environment, flora can quickly transfer to flora, fauna, and humans. Environmental PFAS contamination, including of ground and surface water, has been attributed to hundreds of former and current DoD facilities with the historic use of AFFF, as is shown here on this map. For an example of this contamination in river systems, I've zoomed into my region, which is Southeast Pennsylvania and the lower Delaware River. In the inset graph here, you can see an example of concentrations of one particular PFAS compound in both water and fish tissue in the mainstem Delaware. Moving from right to left, you can see this represents the reach from Trenton down past Wilmington. And you can see in this reach a sharp rise in the concentrations, PFAS concentrations in both water, represented by the bars, and fish tissue, represented by the dots, as a result of integrated inputs of PFAS from tributaries draining DoD facilities with historic AFFF use. This includes Willow Grove and the Joint Base McGuire-Dix Lakehurst complex, as well as several major manufacturing or former manufacturing plants and non-point sources due to wastewater treatment plants. Downstream of these sources, water concentrations quickly decline, but fish tissues still remain elevated. And I acknowledge all this data comes from the Delaware River Basin Commission. Because of the concerns and the prevalence of PFAS in the environment, it is critical for the DoD to understand the ecological as well as the human health risks associated with the presence of PFAS in aquatic environments. We need to quantify the extent, the pathways, and the rates of PFAS uptake and bioaccumulation in order to inform accurate quantitative prediction of risks and evaluate potential pathways for human exposure. So, given that, what do we know about PFAS in aquatic environments, and what are some of the outstanding challenges? In short, we know that the relative distribution of PFAS in both abiotic media and associated biota depends on a multitude of factors, including transfer pathways to tissues, compound-specific chemical attributes, environmental conditions, and much, much more. So to dig into these a little further, I'll start with an overview of transfer pathways. There are multiple pathways by which environmental contaminants, including PFAS, transfer from the environment into the tissues of aquatic organisms. The first is environmental exposure, meaning the incorporation of PFAS through skin or respiratory contact. And this is quantified through bioconcentration and bioaccumulation figures as illustrated here on the right. So in addition to exposure from contaminated water, we also know that some PFAS can partition to sediments, and therefore sediments may act as a sink and a secondary source for biotic exposure. A second major transfer pathway is trophic transfer, and that's exposure through diet or the consumption of contaminated food items. And this is quantified through biomagnification factors or trophic magnification factors. The third pathway, which we know a little bit less about, is the transformation of precursor compounds, including, for example, the PFAS precursor at FOSA and FOSA. We also know that PFAS transport through ecosystems is not a one-way street. Excretion returns some assimilated PFAS back to the environment where they're then available for reassimilation. Illustrating the confounding effects of these many overlapping exposure and transfer pathways, it's not surprising that PFAS concentrations in aquatic organisms have been observed to vary widely based on location, organism trophic level, habitat, and habitat, including both benthic, sediment surface, or water column, for example. So there are several key biorelevant properties of PFAS specifically that we also need to consider. Not all PFAS compounds act the same, and this may impact or affect both the bioavailability of these different compounds in the environment, as well as their relative transfer to organisms. We know that the physicochemical properties of PFAS vary with functional head group and chain length. For example, PFAS sorption to sediments increases with chain length, and as a result, sediments can act as a sink for longer chain perfluoroalkyl acids or PFAAs in aquatic ecosystems. The same has been shown for biota, with long chain PFAAs being shown to bioaccumulate as well as biomagnify. The plot here on the right shows an example of how the bioaccumulation factors for 3 specific PFAAs vary as a function of trophic level in aquatic invertebrates, and this is data from a recent paper by Koch et al. You can see here the bioaccumulation potential increases with increasing chain length across these 3 compounds. The influence of chain length gets a bit more complicated when you look at biomagnification factors specifically. Here we've seen that long chain PFAAs have been found to both increase and decrease with increasing chain length. So clearly we still have a lot more to understand. Besides chemical structure, we also know that the environmental mobility and bioavailability of PFAS is influenced by the environment, particularly the solid phase characteristics and solution chemistry, thinking of things like temperature, cation concentration, or sediment organic carbon concentrations. To date, most of the biological studies have predominantly focused on PFAS and PFOA, although this is slowly changing, but still we understand a lot less about other chain links or PFAS classes. And therefore we need more data on compounds other than just these 2, both as individual compounds as well as in mixtures. So what do we need to consider when we're quantifying and predicting both the mechanisms and rates of bioaccumulation of PFAS in aquatic food webs? Well, we know that bioaccumulation of PFAS is influenced by the PFAS chemical structure, as we just saw, especially for chain length, and PFAS concentration overall. We also know that environmental chemistry has an impact, as does the organisms that are being affected, particularly their habitat and their degree of sediment or water association, and also the organism's feeding behavior, whether they're, say, filter feeders, foragers, or predators. It's important to note here that PFAS have been demonstrated to accumulate more in protein-rich tissues and be possibly lipophilic, which is why the sort of traditional lipid-based partitioning models used to predict bioaccumulation of PCBs and other neutral organic compounds are not really appropriate when predicting PFAS bioaccumulation. What has been shown to be somewhat helpful is looking at PFAS accumulation profiles, as these can help reveal potential different routes of exposure with dissimilar association patterns, accumulation patterns being observed amongst different trophic positions. When we focus at biomagnification specifically, things get a little more complicated. There's a wide range of trophic magnification factors that have been observed across different food webs that illustrate that PFAS biomagnification is particularly hard to predict. There are a couple of reasons why this may be the case. For one, in biomagnification, we still have to account for PFAS compound structure and concentration. And we also need to disentangle this trophic pathway from exposure pathways. Here again, transformation of precursors can definitely complicate things. The apparent biomagnification potential of PFAS and PFCAs may be inflated due to varying abilities of different organisms to metabolize the precursors of these compounds. Furthermore, with biomagnification, we also need to consider methodological error in how the biomagnification or trophic magnification factors are calculated. For example, whether they're whole body or composite or specific tissue based, and whether they account for differences in biomass and different transfer efficiencies. So when quantifying bioaccumulation and biomagnification, we also have to consider the experimental system in which these measurements are made. Some studies have suggested that laboratory-based predictions of bioaccumulation may not be that applicable to field-based measurements. We have a lot of lab studies to date, but there is a limited and yet now growing number of field studies, specifically those in freshwater lotic ecosystems that are exploring multiple trophic levels. So, given this background, and from my personal perspective, there are sort of 4 critical unknowns that we really need to understand with respect to uptake and accumulation of PFAS in aquatic systems. The first is to understand the environmental factors that impact PFAS bioavailability and uptake, particularly the effect of water and sediment geochemistry. Second, we need to identify and quantify the pathways and rates of PFAS uptake and accumulation in order to disentangle and determine the relative importance of the different exposure and transfer pathways. This is especially important when considering mechanisms of accumulation for higher trophic level organisms, for example, fishes that may be consumed by humans. The third critical unknown is the influence of PFAS chain length and head group, and in particular, quantifying more than just PFOA and PFOS in order to determine PFAS compounds that are of the highest concern. And fourth, we need to get a better understanding of the effect of precursors and their transformation. So these sort of data on these unknowns in combination with the development of predictive models and toxicological information is what will allow us to assess and predict the trophic levels, species, habitats, and sites that are at highest risk. Addressing these critical unknowns will require robust field data that we can then compare to pathway-specific results from controlled experiments. And this must be done in environmentally and DoD relevant concentrations and conditions. So these critical unknowns informed the objectives of our ongoing SERDP-funded study with the objective of improving our understanding of the pathways, rates of uptake, bioaccumulation, and biomagnification of AFFF attributed PFAS in stream ecosystems. We are conducting 3 coordinated studies, which include a field study and 2 controlled experiments, and across all of these, analyzing for at least 14 different PFAS compounds, representing a variety of chain lengths, 2 polar head groups, and some that are not fully fluorinated. The 4 guiding questions that bridge these 3 sort of connected studies are as follows. First, what is the extent and pathway by which different PFASs bioaccumulate and or biomagnify within stream food webs exposed to AFFF attributed contamination? Second, which PFAS biomagnify, and does this biomagnification correlate with chain length? Third, how does the biochemistry or biogeochemistry of sediments and water affect the bioavailability and bioconcentration of PFAS specifically to lower trophic level organisms? And fourth, what are those relative mechanisms of PFAS contamination in freshwater ecosystems? In particular, do higher level trophic level organisms accumulate PFAS more via the consumption of their diet, so lower level organisms, or more from PFAS exposure from the water column or sediment? So next, I'll introduce our 3 studies in a little bit more detail. So the first is this comparative field study, which is led by myself, with the objective of assessing PFAS bioaccumulation and biomagnification across the trophic levels, or all trophic levels, of a stream exposed to AFFF attributed PFAS contamination. We will be collecting samples to measure PFAS concentration in both the environment, including surface water, pore waters, and sediment, as well as in the aquatic food web, representing biofilms, aquatic plants, macroinvertebrates, fish, maybe even some riparian species, basically everything we're able to find while we're out there. Based on these measurements, we'll be able to quantify a couple of things. First, the extent of PFAS contamination based on these absolute PFAS concentrations in the various environmental and food web compartments. We also hope to use the relative concentration profiles of various PFAS compounds to help reveal different routes of exposure. But it's important to note here that in our field study, we cannot explicitly distinguish transfer pathways or rates, which is where the controlled experiments then come in. Another thing we're looking to quantify is using the biota or environmental biota concentration ratios to quantify bioaccumulation factors. In addition to PFAS, we will also be analyzing our samples for stable carbon and nitrogen isotopic composition in order to determine food web structure. With this food web structure, we'll be able to quantify biomagnification via trophic magnification factors, which is essentially the regression between PFAS concentrations and trophic level as established with those stable isotopes. Trophic magnification factors can be represent the average diet to consumer transfer of a chemical, or PFAS in this case, through the food web, and can better account for differences in biomass and contaminant transfer efficiencies than, say, traditional biomagnification factors or predator-prey ratios, which really can only be applied to individual species and have been shown to be highly variable between predator-prey combinations. As illustrated by both these kind of concepts figures on the right, we are particularly interested in the effect of organism habitat, particularly the degree of sediment association, and with PFAS chain length on our field study results. The field site that we are going to be using for this study are a series of streams draining the Horsham Air Guard Station and former Naval Air Station Joint Reserve Base of Willow Grove in Southeast Pennsylvania. We've selected 3 contaminated and 1 reference sites, which were selected or which are co-located with NAVFAC water and sediment sampling sites. These sites represent a range of different stream sizes, PFAS concentration, and transport pathways from the AFFF source. Unfortunately, I don't have results to show today because sampling was delayed a year due to COVID, but we will be able finally to begin next month, so do stay tuned. The second study, which is led by Drs. Erica McKenzie and Chris Salas, is a controlled geochemical study. Here, the objective is to quantify the effect of key physical and geochemical factors on PFAS bioavailability and uptake rates from both sediment and water to select lower trophic level organisms. The objective here is also to target DoD and environmentally relevant conditions. The geochemical effects that we'll be considering include solution chemistry, organic carbon concentrations, and temperature, and the results will be used to quantify both bioconcentration factors as well as bioaccumulation factors, which we can then compare to soil water distribution coefficients. We have been, over the last year, running various test experiments to understand things like the time needed for the system to reach equilibrium, as well as how to reduce variables within the experiment while still accurately reflecting field conditions so that we can compare these results to our field results. We've successfully demonstrated that we can use tanks of mixed organisms and are focusing on a combination of species that represent both a range of sediment association and feeding behavior, including algae, mussels, snails, and chironomids. The PFAS loading experiments themselves are just getting started after, again, another COVID delay. So the third study is a controlled trophic study led by Dan Spooner, Dr. Dan Spooner. The objective here is to quantify the rates of PFAS transfer along controlled primary as well as trophic pathways to aquatic species representing multiple trophic levels. The controlled pathways that we'll be targeting include uptake, trophic transfer, and excretion. So with this experiment, we are taking a stepwise approach, focusing first on how the source of how the source of environmental exposure influences PFAS loading to primary producers, such as biofilm, as well as primary consumers, such as benthic and surface invertebrates. You'll note that this is similar to the previous experiment or study, the geochemical study, and this is deliberate in order to help comparison of the results between these two. For the second step, we'll be adding in trophic transfer to evaluate specifically how PFAS from the base of the food web, i.e. those primary producers and consumers, is transferred up to higher level organisms. And then the third step will be isolating the effect of excretion specifically from these higher level organisms to see how this excretion influences the distribution of PFAS in the water column and in the sediment. This particular study is still in the pilot testing phase, but we anticipate it'll go much faster due to the lessons learned from the geochemical study that it builds upon. So across all of our 3 experiments, we're using a common chemoinformatics approach as a hypothesis and integration tool. Specifically, we're utilizing the OPRA, or Open Relationship Activity Model, which uses molecular features to predict the fate and transport of different compounds, PFAS compounds in this case. Using this model, we'll try to forecast which PFAS should be associated with the different ecosystem compartments. And then we'll use these forecasting results to help with the interpretation of our experimental results, focusing those experiments and with information transfer across them. We're also interested in validating the OPRA model utility for forecasting PFAS risk, as we're certainly aware of some complications associated with key assumption of this sort of type of modeling approach. So in conclusion, the quantitative prediction of PFAS risk to aquatic ecosystems requires being able to disentangle the multiple factors that affect PFAS bioavailability and accumulation. Our ongoing study takes a multi-scale experimental approach to trying to solve this, using comparison of results across scales and system complexity from controlled experiments targeting specific pathways and conditions up to the full food web of an AFFF impacted site. We'll also be using that chemoinformatics approach to help with integration. Please do stay tuned for the experimental results over the next year as they start to come in. The benefits to the DoD are several. Our results will contribute basic data that's needed to help support the quantitative prediction and assessment of ecological risks of PFAS to aquatic wildlife and humans at DoD AFFF impacted sites. Specifically, this includes data identifying things like the PFAS compounds of highest concern, the trophic levels, habitats, and species that appear to be at highest risk, the environmental conditions and therefore sites that may be at highest risk, potential pathways to human exposure, and quantifying those partitioning factors for a range of different PFAS compounds, including bioaccumulation, bioconcentration, and trophic magnification factors. We're also, another benefit will be the validation of this chemoinformatics approach for forecasting, specifically the OPRA model-derived chemical properties and how they can be used or not to potentially evaluate the fate of PFAS in the environment. So with that, I want to acknowledge a long list of collaborators on this project, my 3 fellow co-PIs, our 2 PhD students, and a whole series of key collaborators at our various organizations. With that, I'll be happy to take any questions. If you're contacting me afterwards, I just request if you could please use email, as I'm still not regularly in my office and therefore answering that phone number. So with that, Rula, I will turn it back over to you.
Yeah, thank you so much, Marie, for a wonderful introduction to the topic. We've received a lot of questions, and as time allows, we'll start going through them in order here. This is a question from the Massachusetts Department of Environmental Protection. As you do the work that you propose and to answer the or respond to the 3 objectives that you laid out, are you planning to use the top assay on any of your samples?
Yes, that is definitely in our plans.
Wonderful. This next question is from the Norwegian Geotechnical Institute. Would you please describe a little bit more the approximate PFAS mixture composition in your field study?
In our field study, yes. So that is, it is representative AFFF, so the main source. If I recall, I think we have around 60, or in the water at least, it's about 60% PFOS, so that's the dominant form we have, and then about 20% PFHXS, and then a smaller fraction of some of the other head group and chain links represented. Of course, the question, that's one thing we're still sort of refining, is the exact mixture we'll then be using in our controlled experiments. The hope is to try and create a mixture that's similar to this AFFF based source, again, so we can sort of have equal comparison of results between the 3 different studies.
Thank you, Marie. This next question is from EPA. What analytical method are you using for tissue analysis, and does this method include isotope dilution?
Yes, we will definitely be doing isotope dilution, and now I am going to naturally blank on the methods. Give me 1 second. Oh my gosh. Sorry, LC-QTOF-MS is going to be our main approach in, yes, including isotopic dilution. And then I think, as someone said earlier, the last question, we will be doing for select samples that TOP assay for the PFAA precursors as well, and potentially also some non-target screening.
Great, thank you. This next question is from Exponent. It seems like biomagnification via the food web can be confounded by fish age, type of food, and exposure history to water. Is biomagnification a process established for PFAS, or is it more of a hypothesis?
Ooh, interesting one. So I would say it's tentatively established. Enough studies have shown that it definitely appears to be occurring, at least with PFOS and PFOA. I think, yeah, the question here is truly what is biomagnification as opposed to all these other different routes of exposure. And that is really the trick. And as I said, the field study, and in all field studies, that's not something you can determine. You just know that PFAS have gotten it into an organism in some way, but not how. And so this is where these controlled studies, and to the degree that one can, even in a controlled study, truly isolate sort of a pathway, or a trophic pathway in this case, is really the only way that we'll be able to really affirm whether the data that's currently is out there sort of is correct in showing that appears, yes, biomagnification is occurring, not just hypothesized.
Thank you so much. And this next question is from North Carolina State University. Why are lab models of bioaccumulation thought not to be an accurate reflection of what's happening in the environment? Can you elaborate just a bit on that?
Yes. So there are a couple of different reasons. The first is that, well, first can be, let me get my thoughts straight. Well, yeah, timescales are definitely one that we need to consider. So for example, in field studies, obviously the field system has presumably come up to equilibrium. Of course, if you have variability of PFAS concentrations in the environment, how that aligns with the timing of uptake or excretion is still somewhat unknown. But still, we'll sort of make the more general assumption that these systems have come to equilibrium. There's also in a natural field system a huge range of variables that we either know about or maybe don't even appreciate. And so I'd say these are the two main reasons. Those sort of multiple variables and the timing is why it appears that field and lab comparisons are not fully aligned. It's not that they can't be, it's just that we're missing something in that comparison.
Great. And this next question is all the way from the University of the Philippines. Can you elaborate a bit about the role of microbial communities in PFAS transformations and whether you're going to be considering that in your study?
Ooh, that is a great question. So we are not, unfortunately, going to be considering them. We will do a little work with biofilms, but not really analyzing the microbial components of those. I will say for that reason, I am not prepared to answer, but I would be happy to follow up sort of more about the microbial interactions and how that might play into this. There's definitely some interesting studies that I've seen that do account for various microbial components. So that's definitely something that needs to be addressed, just not in our particular study.
Great, Mary. We're getting so many questions.
Yeah, I know, I'm terrified.
No, you're doing great. The next two that I'm going to ask you are from two different parts of EPA. The first one is, are you considering uncertainty and sensitivity when using your methods?
Yes, definitely. So again, I won't go into details. That's one of the things that I'm certainly focusing on a lot right now as we prepare for the field study, trying to find sites that we knew we could get quantification above detection limits was certainly a concern. There's been a lot of field studies out there where they either find PFAS in water, but not in biota, and vice versa. And then yes, how to account for, and there are a number of different sort of suggested approaches for accounting for the precision of method results, as well as what to do when you don't get methods above PFAS concentrations above detection limit, how to account or not account for that in the calculations of the various factors and interpretation of the result. So I will say yes, in short, and a lengthy, I'll be happy to follow up if you have particular suggestions.
Thank you so much for that. And I'm sure you'll be describing all of this in your final report, and that link to your webpage where updates on your progress will be made is available in the slides if all of you guys are planning to download them. Another question from EPA, will protein content be measured in sediment and tissue samples to correlate with PFAS concentrations?
In some of the studies, yes, we are intending to do that. It wasn't in our original design, but having gotten feedback over the last year or so, we do think that is important to contribute. It'll just be a matter of, I'd say, budget and feasibility, or probably where possible is preserving sample so that based on the results that we see, we can go back and add in some of these additional analyses to try and refine the interpretation of the results that we're seeing.
Very exciting. All right, we'll take a few more questions here and then move on to the next speaker. This is a question from the University of Guelph. What is the mode of PFAS for aquatic species? Can you go in a little bit more detail about this?
The mode, I'm not sure I follow, sorry. In terms of I guess they can't answer because it's in a chat. Can I defer that one and follow up with again afterwards, Rula?
Yeah, the mode of action. But if you prefer, we'll provide you with the person that asked that question. You could follow up with them via email.
Great, thanks. I'm drawing a blank.
No worries. One more question from EPA. Are pore water concentrations being considered in your study design?
Absolutely, yes. So we will be looking at surface water. We'll be looking at shallow benthic, so stream bed pore water concentrations. We're also hoping, we haven't tested it yet at the specific sites, but to be able to take some shallow groundwater samples using some multi-level piezometers as well. So trying to characterize, not what just is in the stream, but truly what's in the pore water, how that compares to the sediment concentration, and how does that potentially compare to the source, which is the groundwater sort of upwelling into these streams.
Perfect. And the last question, also from EPA, are you gonna try and close the mass balance in your study? And are you considering the reaction of PFAS with other compounds in the environment in your experiments?
We are going to try, yes. And I'd say that's part of the study design and the analytical design that we're currently debating is making sure to the extent we can that we're able to do that. So I'd say further details coming on that, but very good question, definitely.
Fantastic. Really nice job.
Oh, thank you, Rula. I get off the hot seat.
Yes, for now. We're going to come back to you later at the end of the presentation. Again, thank you, Marie, for a wonderful introduction to this topic. And it is now time to transition to our second technical presenter, Dr. Jamie Suskey, who's a senior scientist at EA Engineering, Science and Technology in Hunt Valley, Maryland. Jamie's current area of research focuses on ecotoxicological effects and risk assessment of contaminants of emerging concern, including PFAS. And in addition to assessing PFAS risk to threatened and endangered species, Jamie is also leading research efforts on the ecotoxicity and biodegradation of fluorine-free replacement firefighting foams. Jamie earned a bachelor's degree in biology at Towson University, a master's degree in environmental toxicology from the University of Maryland, and a doctoral degree in ecology from Texas Tech University. Jamie, we're happy to have you. Please proceed. And you're on mute. If you could please unmute yourself. Jamie, you're still on mute.
Hey there, can you hear me now?
Yes, wonderful. Please
Thank you. Thank you. So thanks, Rula, for the introduction. Sorry for the trouble unmuting myself. Thanks for the support there. Today I'm going to talk about our project that focused on investigating the potential risk of PFAS exposures to listed species on DoD sites. And this is a project that we have recently completed. So the flow, excuse me, the flow of this talk is going to follow the agenda that's outlined here, starting with a problem statement that Marie has already set up for us, which is great. Thanks, Marie. Following with objectives, methods, results, risk framework, some conclusions, and then some benefits of this work to the DoD. So DoD has wildlife habitat that is impacted by per and polyfluoroalkyl substances as a result of the use of AFFF, aqueous film forming foam. DoD is also home to many wildlife species, including many listed species. The goal of our project was to develop a spatial approach for assessing PFAS exposures to listed species and wildlife in general. Secondly, to develop a risk framework for two species, the bald eagle and the northern pine snake. Our objectives were twofold. We aimed to develop a spatial exposure assessment of PFAS exposures through determining the co-occurrence of PFAS contamination with T&E species on the DoD sites. This results in a ranking of species based on proportion of impacted habitat. From here, we selected the two species that I mentioned previously, the bald eagle and the northern pine snake, to develop our probabilistic risk assessment. So spatial exposure assessments are site-specific. First, we determine what species are likely to be present using the Integrated Natural Resource Management Plan, or NRAM, and we supplement that with logs from the federal, state, and county listed species. Simultaneously, we acquire the GIS land cover layers from the installation themselves or from federal partners using data from the USGS, as well as the US Fish and Wildlife Service. We also acquire any landscape layers that the state agencies may have. So once we know who's there, who being the species, we can then look into their life history traits using the literature. And so this would mean that we want to figure out what are their habitat requirements. So where do they like to hang out? What are their home ranges? How far do they move off of their habitat? And then what are their dietary preferences? So for example, we can look at the Vesper sparrow here, which is a state endangered bird in New Jersey. It prefers an open area or field, and it feeds on seeds, waste grain, insects, and spiders. So it has an omnivorous diet. So after we figure that out, or simultaneously, we can also collect data on where the AFFF release has occurred on the installation. And we obtain this from the installation themselves with the preliminary assessments and or site investigation reports, where they have details as to where the AFFF has been historically released. So this is an example that I'm gonna go through briefly using Lakehurst. So this is a portion of the Joint Base McGuire-Dix Lakehurst, which is a very large installation. And so we can only show Lakehurst here to get some definition of features. And so, for example, we've taken the northern pine snake and done a literature search on the habitat requirements and its dietary preferences and where we expect to find this. And we then marry those habitat requirements with GIS landscape layers. So excuse me, at this point, I want to highlight that we did this, although I'm just showing you Lakehurst, we did this for the entire installation, as well as two other bases. One is the Naval Air Station Patuxent River, and then the third would be the Barksdale Air Force Base in Louisiana. So after we pull this together, I can now show you the spatial exposure assessment for the northern pine snake on Lakehurst. So this is, again, specific to Lakehurst. So what we're looking at here is the light green patches are going to be the optimal habitat in which the northern pine snake will hang out. The black hash marks are the range, or how far we would expect the northern pine snake to travel off of the optimal habitat. And then the red, orange, yellow polygons are the reported AFFF release sites. And the different colors indicate the intensity of the AFFF that was released at those locations. So for instance, a red polygon would indicate a repeated release from something like a fire training area, where a yellow polygon would indicate an isolated release. So for instance, putting out a fire from a crash. And then while we know that AFFF does not stay at its release location, and now I'm going to transition to incorporating PFAS as part of this, because PFAS come from the use of AFFF. And so we know that PFAS and AFFF migrate from these points of release. And so the migration pathway is highlighted here in the purple hash marks. And so the pathway is based off of topography, the landscape features, as well as surface water and groundwater directions. And so now what we see is a quantifiable way that we can assess the overall impacted habitat, looking at the overlap of the either AFFF release sites with habitat, or looking at the migration pathways with the habitat. And so again, using Joint Base McGuire-Dix Lakehurst as my example, here is the overall percent land cover that is estimated to be impacted by PFAS on JBMDL. And so this is the overall percent of all habitat occupied by all of the listed species that we have for that location. And so we see that McGuire has comparatively greater percent estimated impacted land cover compared to Dix and Lakehurst. And this is likely due to the fact that McGuire is one of the smaller bases of the three, especially compared to Lakehurst, which is very large. So this can be broken down by taxa and by taxa and diet, as you see here, and then even further into individual species. And I'll show you that in just a bit. And so here, if we look at the percent impacted habitat for insects, we see that it's pretty high in comparison to the other species that I'm showing you. And the inverse is true for the bobcat, which has a, sorry, I failed to mention that insects have a very small home range, which is likely driving their percent impacted habitat up. And so then the inverse is true for the bobcat, which has a very large home range and thus has a larger feeding area. And so then their percent impacted habitat is lower. So avian impacted habitat is smaller among the omnivore and carnivore species, or is similar among the omnivore and carnivore species, and the insectivorous avian species matches the percent of impacted habitat for insects, as you would expect. So looking at the herpetofauna, we see most habitat is impacted again at McGuire, with the herbivore reptiles having the least estimated impacted habitat compared to the other herpetofauna taxa. So moving on to looking at the species-specific estimates, we can see which species may be contributing the most to the taxa dietary percentages. So for instance here, looking at the percentage of carnivore reptiles, which is represented by timber rattlesnakes, corn snakes, and northern pine snakes, we see that the majority of the impacted habitat is being driven by the northern pine snake. So in looking at the habitat assessment, we can see, so this is for McGuire, we can see that this is largely driven by this one area of habitat identified right here. And therefore we carry the northern pine snake through to the probabilistic risk assessment. So here is the model construct. And from the literature, we were able to obtain metabolic demands of the northern pine snake to determine food ingestion rates. So I'm going to start up here. Then a snake will feed within a habitat, and this would thus dictate the PFAS exposure within the food item. And then we compare these exposures to our toxicity reference value, or our current working toxicity reference value. The probabilistic construct of this model is over here on the right. Within the model, a snake may select, so select in quotes, a small or large prey item. This in turn can influence the snake's feeding frequency. So for instance, if a snake selects a larger prey item, its feeding frequency will decrease. And so the inverse is true. If a snake selects a small prey item, the feeding frequency will increase. Finally, the PFAS concentration within the food item varies depending on where the prey item was captured. So for instance, if the snake captured a mouse in an area that is heavily impacted by PFAS, then the PFAS concentrations of that prey item would be higher. If a snake captured a mouse or a prey item in an area of low concentration of PFAS or no measurable PFAS, then that dietary item would have less PFAS within it for exposure. Okay, so what this ends up looking like, this is a snake, an individual snake simulation over the course of 90 days. And so to orient you to this, because I'm going to show it to you again, we have days on the x-axis here. So this is going through 90 days. And then on the y-axis is the exposure. So this is milligrams of PFAS per kilogram of body weight per day. And so what's important to take away from this is that each peak you see here is showing you a higher PFAS concentration within the prey item that the snake has consumed. And so you see that there's a flat line towards the bottom of the x-axis, or sometimes maybe a little blip. And that doesn't necessarily mean that the snake hasn't eaten during that time, but that the snake just did not consume a prey item with measurable PFAS concentrations. Okay. So looking back at McGuire, where we have approximately 13% of the snake's habitat impacted, taking the mean PFAS exposures from 1,000 simulations, we end up with, right here, we end up with a mean PFAS exposure of around 0.01 milligrams per kilogram per day. Currently, Our toxicity reference value, which is derived using Brown and Knowles from Dr. Solis's lab, is at 0.17 milligrams per kilogram per day. We can see that our y-axis actually doesn't even extend up that high. So we can say from our model that it does not exceed the working toxicity reference value. And thus this indicates that there may not be a concern. Okay, transitioning to our next species, the bald eagle. This is the habitat exposure estimate for fish-eating birds. Right here is contributing a great deal to the overall avian carnivore percent impacted habitat. And so both bald eagle and osprey are similar because they have similar dietary preferences. However, the bald eagle does supplement with prey items from the terrestrial habitat. So we chose the bald eagle because it is found on many DoD installations and also found on all 3 installations that we studied. Additionally, I recognize that it is no longer on the endangered species list. However, it is still a protected bird. And so for these reasons, we have carried this species through to the probabilistic risk estimate. And so moving off of JBMDL and onto Barksdale, we chose Barksdale Air Force Base to build this probabilistic risk model because we have published fish data from Lanza et al. 2017. And we know that the Cooper Bayou is an impacted site, and those are where our data are from. It's kind of hard to see, but there are these little yellow dots here with the measured fish tissue concentrations along with water concentrations from the Cooper Bayou. Here's Cooper Bayou that I'm drawing the arrow along. And so this figure for the bald eagles is similar to the one I showed you previously with the northern pine snake, where we have the light green patches indicating that this is optimal habitat for the bald eagle. And then you can see the black hash marks all over the installation because the bald eagle has a very large home range. And then again, these polygons, color-coded polygons, are indicating areas of AFFF release, as well as the purple hash marks indicate the migration pathway that we expect PFAS to travel from those release locations. Okay, so importantly, this model included a function of nest proximity to the impacted Cooper Bayou. So we know that Cooper Bayou is impacted because we have those measured data, and this is not only from our habitat exposure assessment. We also focused on the eaglet as our life stage because we believe that this was a sensitive life stage, more sensitive than the adult phase. And so within the model, PFAS exposure was a function of proximity to Cooper Bayou, where we assumed that the nest locations closest to the bayou here will be feeding on fish more frequently with PFAS accumulation from the bayou than, say, nests that are further out here, where likely eagle parents will be selecting prey items from other locations. So here's the model construct for the eaglets. We used allometric equations for food ingestion and growth through time. PFAS and dietary items was from published literature, again, the Lanza et al. In fish. And then we also have some other sources for PFAS concentrations in mammals. The probabilistic components of this model are right down here. So did the parent eagle select a mammal, or did it select a fish prey item for the eaglet? Moving on, was that prey item contaminated? If yes, then a PFAS concentration was selected from a distribution. So this is the same construct for the simulation that I showed you for the northern pine snake, where we have days on the x-axis. So right here is day 0, where the eaglet has just hatched, all the way through fledgling, which is around 62 days. On the y-axis here is the PFAS concentrations that the eaglet would be exposed to. And I just want to note that early on, following hatching, that the eaglets have a very small body size, and they also have an extremely high metabolic rate at this point. And so again, these higher red peaks indicate higher PFAS exposures, and then these low to no PFAS exposures does not necessarily mean that the eaglet wasn't eating, just that there was no PFAS or measurable PFAS within that food item that was brought back to the nest. And so what you see is this toxicity reference value. This is represented by this blue line, and this was established by Newstead et al. This is the threshold where we expect there to potentially be some deleterious effects. And this is set at 0.21 milligrams per kilogram per day. And you can see there are very many red lines that are exceeding that threshold. So overall, using the mean of 1,000 simulations, so again, we did the same thing that we did with the snake, and we simulated this for 1,000 times and found that the threshold was exceeded if 20% or more of the diet had come from Cooper Bayou. And so if this is true, we're not necessarily sure, but this is definitely an approach to explore some of these possibilities. So there are a couple main takeaways from this research. First, that spatial habitat exposure potential can be a useful approach to estimating AFFF and PFAS contaminated habitat, and that we can prioritize species that may have high exposure potential. Secondly, avian receptors may be at risk from PFAS exposures, specifically those with small home ranges and fish-eating birds, especially in sensitive life stages. So some benefits to the DoD is that this provides a really rapid desktop assessment of likely PFAS impacted habitat, and we can address the most vulnerable or identify the most vulnerable taxa and or species. Also, that probabilistic risk assessment tools are available, and we can adapt these to other species or wildlife taxa of concern. So I want to acknowledge a few of my colleagues and collaborators that contributed greatly to this work. At EA Engineering, we have Mark Druve and Eric Yan, who are our GIS gurus, and Kat Kearney-Chipman, who was my naturalist support and listed species expert, as well as Dr. Christopher Solis, who developed all the risk modeling efforts for this. And then some of our resources that we use to develop the models can be found here. And with that, I guess I'll turn it over to Rula, but we do have our final report published and is available online, so always feel free to check that out.
Thank you so much, Jamie. And that report is up there right now, and it's available for download at no cost, as are the slides for the presentation, and the video will be available in about a week. So let's start getting to some of the questions that have come in, Jamie. The first one is from EPA, specific to Lakehurst. Does Lakehurst also have habitat and occurrence of the Vesper sparrow specifically?
Yeah, so while I recognize that I use that as an example, there is habitat, and in the final report, if you wanted to specifically look for that species, that information would be available. And so we do provide that within the report through many tables, so you can find that there.
Great. This next question is from the U.S. Army Public Health Center. Is your estimate of PFAS, in your estimate of PFAS mobility layers, how are you determining where the plume or distribution ends if PFAS are resistant to degradation?
Yeah, that's a great question. And I think that that's something that we could definitely continue to explore within that model. So ultimately, our PFAS migrate following surface water and groundwater directions, and then they terminate at a surface water body. So that's the termination point. But I recognize that there are geophysical and geochemical constraints that influence mobility of PFAS within the soils, and we have not accounted for that yet.
Thank you. This next question is from the Massachusetts Department of Environmental Protection. How did you decide to model ingestion over a period or a window of 90 days? Is that a normal window, or is it short, or is it long? How did you get to this number?
This is for the northern pine snake, and so that's a great question, and that's one of the assumptions that we've made. So I think that is something that we could continue to refine and play with if there are other data that are available. But currently, our TRV that we have that we're using is from a 90-day study.
Thank you so much. This next question is from EPA. Were PFAS body burdens of the pine snake considered in food chain modeling to upper trophic level receptors that may feed on these snakes?
No, it was not, and we don't have those data.
Thank you. Another question from a different part of EPA. Are your impacted habitat percentages expressed as % of the total base or as % of the total habitat for the species within the base?
Yeah, that's a great question. And so looking at the overall % of impacted habitat, the percentages are based off of the available habitat that is appropriate for the total cumulative species, as well as for individual species. So it's a function of the available and appropriate habitat for those species.
Thank you, Jamie. This next question is from the West Virginia Department of Environmental Protection. Given that a lot of the sites that we're evaluating here, there are multiple contaminants beyond PFAS, did you assess the potential cumulative impacts of other contaminant releases at the sites that you studied?
So I guess, I mean, no is the answer. But you know, that would be hard to incorporate in our probabilistic risk models. I mean, as maybe I didn't point this out, but I focused on one PFAS within our probabilistic risk models, as it is hard to deal with mixtures of PFAS together, much less build in other contaminants at this point.
Thank you, Jamie. This next question is from the Norwegian Geotechnical Institute. Are toxicity reference values for these species mainly established for PFOS, or do they exist for other PFAS as well?
So the ones that our toxicity reference values are for PFOS, and specifically what I presented here are, but then The Solis lab at Tufts University is working towards developing other toxicity reference values for the reptile model, as well as Dr. Anderson at Texas Tech University is working on developing some and helping add to that literature for the avian species with PFAS and other PFAS.
Thank you so much. This next question is from the University of the Philippines. Do the seasons affect PFAS concentrations, especially with regards to the behavior of animals during these seasons, like the northern pine snake?
I'm sorry, can you ask that question again?
Yes. Do the seasons affect PFAS concentrations, especially with regards to the behavior of animals like the northern pine snake?
Thanks. Sorry. I appreciate that. Yes, so they do for sure, especially with the northern pine snake, where the northern pine snake really hangs out in the hibernacula during the colder seasons, and so it is not feeding during that time. And so yes, seasonality does play a role in feeding frequency for that species.
Great. Thanks. What are your future plans for applying and or validating your model at other sites? Is your work completely done, or do you have any follow-on efforts planned?
Well, actually, thanks for that question. I guess I would like to try to apply this to other installations. I think that we have a great opportunity at the moment right now where we can be pretty creative in how we do risk assessment on PFAS, because we're not sort of pigeonholed to the CERCLA process at the moment. And so we can come up with some creative approaches for handling PFAS. And so I would like to expand this and apply it to other installations and other sites if there's interest.
And perhaps there's some synergies with some of Marie's work. This question actually came from her. So excellent. All right. An anonymous question: how do you account for potential PFAS uptake from surface water within the probabilistic risk assessment model?
So like incidental ingestion, I guess, is what they're leading towards. And if so, we don't have that in the model, but we could build that in if we that could be an addition to help refine it.
Thank you. And perhaps one last question before we pull Marie back in. What were the methods used to determine detectable amounts of PFAS trapped in the bald eagles and pine snakes? That is, how did you know when they consumed the meal and how much PFAS was in that meal?
Yeah, so those were part of the probabilistic components of the models, and so that would vary by simulation, and so it ultimately it's going to be driven by the metabolic needs of the individual species. So how many calories does the snake need per day distributed over the course of that time frame, and same thing for the eaglets.
Wonderful. All right, we got through a lot of questions, both you and Marie, that just talks to the interest in this topic, but I'd like to pull Marie back in and ask you guys some more generic questions about the state of science and where it's moving when it comes to these types of eco-risk issues. So maybe we start with Marie, and thank you, Jamie. What would you characterize as some of the data gaps? You mentioned key data gaps, but if you had to pick one or two that should be focused on for addressing PFAS in the risk assessment process, what would those data gaps be, Marie?
Oh, loaded question. Actually, I'd say it's maybe less of a data gap as a data interaction. And I think what I really liked about Jamie's talk was looking at these predictive models and how do we blend those with the data, the type of data I introduced that we're trying to produce that could be used to either calibrate or validate models like Jamie's using, and then how do we also fold this in with the toxicological information? And so there's a lot of unknowns out there, but as data starts to come in, and there's always more data we're going to want, how do we sort of make the best use of it or create studies that collect the data that can have the most multiple uses for trying to sort of blend all these different directions into sort of the results that obviously are needed on the ground to help for management. It's a little bit of a dodge, but.
That's a good start. Jamie, would you like to add anything to what Marie said?
Yeah, well, I think Marie, I agree with her response. And I also agree with one of the questions that I received about looking at other contaminants, but more so in terms of PFAS mixtures. And that's still something that we're trying to understand. We can sort of get a feel for the toxicity of PFAS, which, as Marie mentioned, is one of the dominant PFAS found in the environment from AFFF releases. But in terms of PFAS mixtures, I think that's still a pretty big unknown, and how we deal with that in the risk assessment framework.
Thank you so much. And in talking about different PFAS, in your opinion, Jamie, is there a specific PFAS that appears to drive more toxicity than others that risk assessors can rely on as a sentinel PFAS, or is risk assessment going to have to address the mixtures issue that you alluded to?
Yeah. So I think the way that the data seem to be unfolding now, I believe that our focus has been on PFAS, not only because it's the dominant PFAS found in the environment, but that it seems to be a driver of toxicity among the PFAS that have been studied, although there's thousands. And so I think that that might be the driver of toxicity at the moment, to answer your first question. The second part of that question is, yes, I think that we need to figure out how to address the mixtures. And I, right now, don't know that PFAS can be used as a sentinel PFAS in that decision making. I think that mixtures is still something that needs to be addressed and is complicating the waters at the moment.
Absolutely. Marie, would you like to add anything to what Jamie just relayed?
I would certainly, well, I'd echo her answer, absolutely. But also say, yeah, I don't think we're anywhere near the point where we can discount mixtures and just focus on some sentinel. I mean, maybe that will be the case down the road, but certainly that data doesn't appear to exist yet. And of course, in sort of natural environments, it's always going to be a mixture. I mean, there's the sort of profile that a lot of us are used to seeing from AFFF, but then there's also, increasingly all the new replacement compounds and other, as Jamie said, those thousands of compounds blending together. And so establishing sort of those effects will really be key, both on an individual and a mixture level, to help better represent sort of the natural environments and where we actually see PFAS.
Wonderful. I'd like to thank you both again for a wonderful webinar. Our next webinar is on Thursday, April 22nd. It'll be in the Resource Conservation and Resiliency Program area. This webinar will focus on research efforts to improve the management and recovery of threatened, endangered, or at-risk species on DoD installations. We'll have 3 speakers, Mr. Patrick Wolf from the U.S. Army Corps of Engineers from ERDC, Dr. J.D. Wilson from the University of Arkansas, and Dr. Brad DiGregorio, also from the University of Arkansas. Registration is open, so please visit the SERDP and ESTCP webinar webpage, and you can register for this and other future webinars through the end of this year. And before we conclude, I'd like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you'd like to refer to them in the future. We got a lot of comments in the chat about the slides, so feel free to go and download them right now. They are available. And at this point, we would really appreciate it if you could just take a quick moment to complete the short survey that will pop up on your screen. This concludes today's webcast. Thank you so much for your attention.